All right, medical students, welcome to today's explainer. If you're prepping for exams and grinding through that CE 2083 syllabus, well, you're in the exact right place. Today, we're unpacking one of the most brilliant and honestly devastating pathogens in human history, the malaria parasite's Plasmodium. So let's just dive right into this and trace its entire pathological journey.
Quick look at our game plan for today. Number 1, we'll meet the malaria parasite. Number 2, the invasion, looking at the liver stage. 3, multiplication in the blood stage. 4, transmission during the mosquito stage. And finally, number 5, targeted treatments and diagnostics.
Let's kick things off with Section 1, Meet the Malaria Parasite.
There are several species out there, but your boards, and your wards for that matter, are going to heavily focus on two main culprits. First up is Plasmodium vivax. This is the most common and widely studied species. It causes benign tertian malaria, which basically means the patient's fever is going to spike every 48 hours. Then you have the most fatal one, Plasmodium falciparum. This one causes malignant tertian or cerebral malaria. You absolutely need to remember this distinction. Vivax is the most common, but falciparum, that's the killer. Quick mental check before we move on. Which species causes benign tertian malaria? That's right, it's Plasmodium vivax.
Moving right along to Section 2, Invasion, the liver stage.
Now, it's fascinating how this initial infiltration goes down. An infected female Anopheles mosquito bites a human and injects sporozoites directly into the blood. But they don't actually hang out there for long. Within about half an hour, they completely disappear from circulation. Why? Because they're on a stealth mission. They rapidly enter the parenchymatous cells of the liver specifically to escape the phagocytic action of our white blood cells. Once they're safely tucked inside a liver cell, they multiply asexually into thousands of cryptomerizoites, eventually bursting the cell to go infect more.
I actually want to take a brief clinical pause right here because this next part is absolutely critical for your practice. Not all of these parasites immediately multiply. Some of those Plasmodium vivax sporozoids actually turn into a completely inactive form called a hypnozoid. Think hypno like sleep, right? This latent stage can just sit there, totally dormant in the liver for months or even literally years. This is exactly what causes a patient to experience a sudden malaria relapse long after they thought they were totally cured. It's what makes Vivax incredibly difficult to completely eliminate.
Okay, time for a quick active recall check straight from your text. Which of the following can start pre-erythrocytic schizogony? Is it A, B, C, or D? Take a second. If you chose A, the sporozoite, you hit the nail on the head. The sporozoite is the infective stage that kicks off that very first liver cycle.
All right, Section 3, multiplication, the blood stage.
Once inside the red blood cell, this crazy cycle of destruction, known as erythrocytic schizogony, really begins. The parasites entering the RBCs are now known as micrometacryptomerizoites, and once they're inside, they undergo these rapid, very distinct morphological changes.
Let's walk step by step through these changes because you'll definitely need to identify them under a microscope. First, it becomes a trophozoid, which is this voracious feeding stage. Then a large vacuole forms, basically pushing the nucleus all the way to the edge, and that creates the classic signet ring appearance. Next up, it develops pseudopodia, turning into the amoeboid stage where it feeds really heavily on the host's hemoglobin. Asexual multiplication then turns it into a multinucleated schizont, and finally the merizoids arrange themselves like little petals on a flower in the rosette stage, right before the red blood cell violently bursts open.
So what exactly happens when that red blood cell bursts? Well, during that feeding stage, the parasite breaks down hemoglobin into globin and hematin. That hematin gets deposited as hemazone, which is this iron-containing toxic pigment. So the really crucial takeaway here is this. When the RBC ruptures, it doesn't just release a bunch of new parasites, it literally dumps this toxic hemazone right into the bloodstream. This sudden release of toxins is the exact biological trigger for those signature chills, the severe shivering, and those cyclical paroxysmal fevers that you're going to see in your patients on the ward.
Let's do another quick checkpoint. This schizont stage of Plasmodium is formed in A, B, C, or D. Give yourself a pat on the back if you selected C. Remember, schizogony, that whole process of multiple asexual fission, happens in both the human liver cells and the red blood cells.
Moving on to Section 4, transmission, the mosquito stage.
Now, during the blood stage, some of these parasites actually don't become schizonts at all. Instead, they develop into male and female gametocytes. When a new female Anopheles mosquito swoops in and takes a blood meal, she sucks up these gametocytes. The temperature drops, which shifts the parasite to a cold-blooded host, and that kicks off the whole sexual cycle right inside the mosquito's gut.
Inside the mosquito's stomach, the male microgamete fertilizes the female macrogamete. This forms a diploid zygote, which then elongates into this highly motile, worm-like structure called an ookinete. Now, get this. The ookinete literally wriggles its way right through and penetrates the mosquito's stomach wall, settling on the outside to form an oocyst. Inside this oocyst is where sporogony occurs, creating thousands of new infectious sporozoids. Actually, fun historical fact for you. It was Sir Ronald Ross who first discovered these oocysts on the stomach wall of mosquitoes way back in 1897. Won him a Nobel Prize.
Okay, one final rapid-fire review for the life cycle. In the life cycle of the malarial parasite, the sporozoites are formed in A, B, C, or D. The answer is A, the oocyst. When that oocyst finally bursts, thousands of sporozoites are unleashed into the mosquito's body cavity, and they migrate straight to the salivary glands, completely primed and ready to infect the next human victim.
Let's bring it all together in Section 5, Targeted Treatments and Diagnostics.
For rapid diagnostics on blood films, you really want to look for the unique granules deposited in the red blood cells. This is a classic board exam topic, so definitely lock this in your brain. P. vivax and P. ovale, they show Schüffner's dots. P. falciparum shows Mahrer's clefts, and P. malariae shows Siemens dots. Memorize these associations, guys. They will absolutely save you vital time in high-pressure clinical scenarios.
When it comes to treatments, they are strictly stage-specific. And this is exactly why understanding this whole host-hopping journey is mandatory for prescribing. Primaquine targets the latent hypnozoids and gametocytes. Chloroquine and artemisinin-based combination therapies, or ACTs, they wipe out the active blood stages. And proguanil attacks the liver schizons. You can't just throw any random drug at this parasite. You literally have to target its current location.
And that brings us to a really crucial dual therapy concept, especially when we're dealing with Plasmodium vivax. If a patient comes in shivering with vivax malaria, yeah, you give chloroquine to clear the active blood stage and stop the immediate illness. But remember those sleeping hypnozoites we talked about earlier? If you stop there, the patient is going to relapse for sure. You absolutely must also prescribe primaquine to eradicate that dormant liver reservoir. You basically have to fight the parasite on two completely different fronts.
This leads to a profound reality highlighted in your syllabus. Quote, why P. vivax will be the last human malarial parasite to be eliminated. Hypnozoite can reactivate later. Unquote. This represents just a massive global health challenge. I mean, how do you totally eradicate a parasite that can simply go to sleep inside our organs, completely hiding from the immune system and dodging most standard blood stage drugs? Time for one last rapid review. Which drug do we use to eradicate that sleeping hypnozoite? Exactly, it's primaquine.
Which brings us to the ultimate question. We've mapped its incredible journey from the mosquito gut into the human liver, through the red blood cells, and back again. But with Plasmodium's complex host-hopping life cycle and ever-growing global drug resistance, are we truly prepared for its next evolution? Keep studying the intricacies of this deadly, adaptable parasite, because one day it's going to be up to you to outsmart it on the wards. Thanks so much for joining me on this explainer, and I'll catch you in the next one.